I'm Cynthia Chang, and I'm presenting this research on behalf of myself and productivity, so how much energy is being captured by the ecosystem. This has relevance to agriculture, many things. Nutrient cycling, resistance to invasion by the community, and the community's ability to be resilient or resistant to different kinds of disturbance or climate change. And when we think about the relationship between diversity and ecosystem functioning, there's a lot of different ways we can measure diversity. So at the community level, people have traditionally thought a lot about species diversity. More recently, we've thought about thinking about things from a phylogenetic perspective, as well as linking it to functional traits.
That's okay.
And then, recently we've also started thinking about genetic diversity within a single population in the community, particularly for dominant species, and how that might play also a role in ecosystem function. So there's a huge body of literature in both ecology and evolutionary research trying to figure out the relationship between diversity and ecosystem function. And so ecologists have come up with many different hypothesized theories for explaining why there might be a positive relationship between diversity and ecosystem function. One of them is niche complementarity, or resource use efficiency, proposed by Tillman and others. And just a simplistic cartoon view, if we have two communities, one community is low diversity, the other community is high diversity. In this case, I've just changed species diversity. You see that the high diversity community, the individuals occupy different niches, so there's more overall resource capture in that high diversity community, and therefore higher productivity. Another hypothesis is the sampling or selection effect. And what this theory says is that if you have a low diversity community versus a high diversity community, it's simply a numbers game. So as you increase diversity, you increase the probability that any one of those individuals or species is going to be particularly highly productive, say, if that was the ecosystem function you were looking at. And that increases the overall community's productivity. And then very similarly, the facilitative effect says same idea. As you increase the number of individuals or diversity, you increase the probability that any one of those species will be facilitative, therefore increasing overall productivity or ecosystem functioning in that community. So there's been a large body of research looking at the relationship between species diversity and ecosystem functioning. Very classic experiment done by Tillman et al. In Cedar Creek, where he manipulated functional diversity and species diversity. He did see that as you increase diversity, you see this positive relationship with productivity. And then more recently, people have started looking at phylogenetic diversity. So as you increase phylogenetic diversity, we also see this increased positive relationship with productivity. But more recently, people have also started looking at genetic diversity. And so study by Kretzinger et al., where he manipulated genotype richness of Solidago. He also saw this increased positive relationship between genotype richness and productivity. And then also in the seagrass community, here what they found was that as you increase genotype richness, you see an increase in the community's ability to resist disturbance, in this case grazing by geese, and also be resilient, so recover and bounce back from grazing. And so together there seems to be a lot of research that suggests that some of these different mechanisms might explain why we might see these positive relationships. However, there are other studies that don't necessarily find these positive relationships between diversity and ecosystem functioning. So a big part of the research now is, I think, trying to go and try and understand why you might not see these relationships when you don't versus when you do. So in a meta-analysis done by Raj Whitlock in 2014, where he summarized all the literature that looked at the relationship between genetic diversity and ecosystem functioning, what he found was that indeed, a lot of times we do see this relationship between particularly genotype richness, but we don't always see a relationship, and this is particularly true in some of more of these neutral measures of genetic diversity looking at relatedness. And I think it's really here, when we don't always see this relationship, where we have to then stop and think about, really, are we understanding this? It's really important for us to try and understand the mechanisms that link genotype to the phenotype, because ultimately it could be at the phenotypic level that might be contributing more to ecosystem function. So, thinking about all of this, more recently people have then thought about, okay, well, what other aspects of genetic diversity might have an impact and be important to ecosystem function? And so Latzel et al., instead of manipulating genotype richness, what they did was they kept genotype diversity constant, but manipulated epigenetic diversity, and you saw indeed this increase. As you increase epigenetic diversity, you see an increase in productivity in Arabidopsis, both grown in competition with other plants and also with pathogens. So this seems to suggest that there's other things about genetic diversity and trait variation that we really need to consider when we're trying to understand the potential importance of diversity on ecosystem function. And so that motivated a lot of our research to stop and think about phenotypic plasticity, which is another aspect of diversity. We know, so phenotypic plasticity is trait variation across different environments. We know that it's very ubiquitous across species. A lot of people have hypothesized that it potentially plays a very large role to the species' ability to adapt, particularly to rapid global change. And in a study done in tallgrass prairie, in a long-term manipulated climate change experiment, they did found that in the dominant species, phenotypic variation due to genetic diversity and plasticity allowed this dominant species to adapt to variable precipitation. So this suggests that phenotypic plasticity could likely not only play a large role to adaptation, but possibly to being important to ecosystem function. So if we think about phenotypic plasticity, this is changes in a particular trait across some sort of environment. You might have an individual or a genotype that's very plastic, and that means that it has more overall trait variation. If you think about it from an ecology perspective, more niche space is being occupied. Versus a low plasticity genotype, there's less trait variation, so maybe less niche space being occupied. And this could be particularly relevant if there's some sort of spatial temporal variability in whatever environment or resource you're interested in looking at. And so we hypothesize that as you increase trait variation, in this case being driven by phenotypic plasticity, we expect to see a positive relationship between that and different kinds of ecosystem functioning. And so for this study, we just focused on productivity. So for our research question, first thing we needed to establish was whether or not the different genotypes we were looking at exhibited more plasticity. In this case, we looked at above ground traits and whether or not these traits were under selection. We, of course, looked at the relationship between genotype richness and productivity in two different environmental drought and non-drought conditions. And then we also looked at the relationship of plasticity at the community level and productivity under these two different water treatments. And so we did this in the context of the large Unpack network. Unpack stands for Undergraduates Phenotyping Arabidopsis Knockouts. This is a collaborative initiative with a lot of different, primarily undergraduate institutions. And the goals of Unpack are to, big picture, try to understand the relationship between genotypes and phenotypes using knockouts, and then also quantifying phenotypic plasticity. So many of you guys I'm sure know Arabidopsis thaliana is an annual plant. Very conveniently has a 6-week life cycle, which fits in great with the quarter schedule semester. So I was able to do this large-scale greenhouse experiment as a cure course in my plant ecology class in 2015. And then I also had a team of undergraduate researchers that stayed on with me and helped with this experiment. So for our greenhouse experimental design, we had 6 genotypes of Arabidopsis. These are natural accessions, and just for simplicity, I just call them A, B, C, D, E, and F. So we first grew these genotypes alone in a pot under high and low water, so that we were able to measure their plasticity. And based on previously unpublished Unpack data, I was able to try and predict, say, the first 3 A, B, C were thought to have low plasticity, but this is something we could confirm with our own data in our greenhouse experiments. And then we also grew the different genotypes and different diversity treatments. So 6 individuals in a pot where I manipulated genotype richness, and then also simultaneously was able to try and manipulate community level plasticity. And again, this is something we can calculate at the end with our, based off of the plasticity levels of the single genotypes. So we had 2 water treatments, drought and non-drought, and then we had 15 replicates of the single genotypes and 30 replicates of the diversity, each of the diversity treatments in each of the water treatments. And our experiment was 48 days. So we cold stratified the seeds. First, they were obtained from the Unpack seed bank. They were cold stratified for a week, 4 degrees Celsius. The students then sowed all the seeds. We did a split block design in the greenhouse. Everything was randomized. Both the pots were randomized and the individuals in the pots were randomized, and I had the students double check each other. We collected specific leaf area, which is a functional trait that is thought to tell us something about competitive ability versus stress tolerance trade-offs. So low specific leaf area says there's low competitive ability, but high tolerance to stress, and vice versa for high SLA. We also collected above ground biomass for each plant and counted fitness, which was just the number of pods for each plant. So first off, do each of the genotypes exhibit more plasticity? So here we're looking at above ground biomass, and we do see variation in phenotypic plasticity across the genotypes. Here I've simply, this is the standard reaction norm, here I've simply just ranked the different genotypes from high to low plasticity. You'll notice for all these different traits, they don't necessarily match the original predicted plasticity rankings. Plasticity was calculated as the slope across the two water treatments. And so, if you remember, the plasticity values were particularly important because they allowed us to calculate the community level plasticity. So in this example, I have the Z treatment, which is all the A, B, C, every single genotype is represented. If a particular individual died, we did not include that in the calculation. We took the average, so the plasticity index per pot was the average of all the plasticities based off of the single genotypes. And then I took the absolute slope. So that way the direction of the change didn't cancel each other out. So really quickly, looking at reaction norms for the other traits, we do see a G/E interaction for fitness, and the plasticity levels are different. They're the rankings for the different genotypes, in addition to a strong genotype by environment. But when we did our selection gradient analysis, we did not find evidence that above ground biomass and specific leaf area were under particularly strong directional selection. So something to keep in mind. So when thinking about the relationship between genotype richness and productivity, what did we find? So overall, when you look at genotype richness on the x-axis, productivity on the y-axis, we did not see a significant relationship between the two, both in drought and non-drought conditions. But then when we look at plasticity of the pot level and productivity, so this is on the x-axis, plasticity index of the pot. This is plasticity of above ground biomass. We also do not see a positive or any sort of relationship between that and productivity for both drought and non-drought conditions. As well, same idea for the specific leaf area, plasticity index using specific leaf area. We also don't see a relationship between that and productivity in either of the water treatments. And then when we look at fitness, plasticity and fitness, we actually see a very weak but significant negative relationship between that and productivity. And this actually, if you stop to think about it, makes sense. You wouldn't expect there to be strong selection for fitness to be very different across the different environments. So something not what we expected to find. So in conclusion, we don't see a significant relationship between either genetic diversity or propensity for plasticity to productivity as we had sought out to test for our hypotheses. And these relationships didn't really differ in both drought and non-drought conditions. But I think this is the first step of, we hope to do several other projects looking at these questions. We really hope to try and think about this in the context of a variable environment, so changing the precipitation watering regime, really focusing on below ground traits which are likely to play a role in drought, and using different knockout lines to try and target genes of interest, and possibly also looking at the impact of community level plasticity on resilience and tolerance to different kinds of disturbance. With that, I'd like to acknowledge particularly Unpack Network and funding resources, our undergraduate other researchers, and the greenhouse staff. And with that, I'm happy to take any questions.